So good afternoon, everyone. I am pleased to introduce Dr. Jena French. Dr. French is an immunologist at the University of Colorado Anschutz Medical Campus in Aurora, Colorado. Her research has focused on defining the immune response in both follicular-derived and medullary thyroid cancers using patient tissues and preclinical models. Her current interests include defining T-cell antigen specificity in patients with thyroid cancer and designing novel combination therapies to improve the response to immune checkpoint inhibitors in patients with progressive disease. Thank you so much. Dr. French.
Thank you. Okay, so as you just heard, my name is Dr. French, Jena French. I'm from the University of Colorado, just down the street. This is our lovely campus. And I'm an immunologist, so this talk may be a bit different than some of those that you've heard. So my focus has been, as an immunologist, on understanding the immune response in thyroid cancers. And more recently, getting into the clinical realm a bit more with a collaborator, also here in Denver, Dr. Brian Haugen, getting into clinical trials that test immune-based therapies for thyroid cancers. And thank you. And specifically, most of what we focus on are immune checkpoint inhibitors, which I'll describe a bit more in detail. I think you've heard about them to some degree in other talks in the last day or two. Okay, so my research is really focused on understanding how we overcome barriers to response for these types of therapies. Do I need to do the arrows? Okay. Okay. Is that me? Oh, okay. Okay, so I wanted to start out with kind of a general overview of the immune response in cancer. We've known for over a century that the immune system plays an important role in controlling tumor growth. And in the last 10 years or so, or actually it's been 20 years, that's crazy, the immune editing hypothesis came out. And that basically states that the interaction between the immune system and tumors occurs in 3 stages. The first is the elimination stage. And this basically is the stage where we think that the immune system is capable of really surveying the body for cancer and is successful in eliminating these early stage cancers. And it's characterized by especially T cells. T cells are white blood cells or lymphocytes. They're also referred to often as CD4 or CD8 cells. And these are the foot soldiers of your immune system. So they can directly kill the tumor. In addition, we have important cells called myeloid cells, which are also white blood cells. You may recognize the terms macrophage and dendritic cells. These are like the sentries of your army. They alert the T cells that something is wrong and that the T cells need to attack, leading to T cell activation. So the tumor, as the tumor progresses, it begins to outsmart the immune system. It starts to change, as you can see here, depicted by the changing in color and form. And the immune system at the same time changes. It becomes edited to some degree and is no longer as good at seeing the tumor. And in the clinically relevant tumors, we have an escape where the tumor has kind of won out against the immune system. At this point, T cells are quite dysfunctional, and the myeloid cells even become dysfunctional. They can suppress the T cell response, and they can even promote tumor growth. And so the goal of immune therapies is really to push us back to this phase where we can have a more activated immune surveillance and hopefully elimination of the tumor. So immune checkpoint inhibitors became big about 15 years ago. And the concept behind these is that there are immune checkpoints, multiple immune checkpoints in your immune system that are expressed to resolve an immune response. So if you have a viral infection, you don't want your immune system to just keep acting and acting and acting because you're going to end up with a lot of side effects. you're going to end up with autoimmunity potentially. And so these are brakes that are turned on by a natural immune system to dampen down that T cell response. So one that is probably most well studied and well known is called PD-1. Now a tumor, again, I said, can outsmart the immune system. So a tumor can actually upregulate the ligand for this molecule. They express a molecule often called PD-L1. And what these immune checkpoint inhibitors do is they are antibodies that interfere with this interaction. So it takes away that inhibition of the T cell response and improves that immune attack. So these therapies are growing rapidly and have been tried in thyroid cancer now as single agents. Unfortunately, the response has not been great as a monotherapy. This is pembrolizumab, which I'll talk about today, also known as Keytruda. In DTC, we saw only 2 patients out of 22, so a 9% response. And in ATC, another drug called spartalizumab that targets the same molecule was tested, and they saw a better response. So 19% overall response, 3% complete response, which is more encouraging. But you'll note that most of the patients failed to respond to this drug alone. So our focus has really been to understand how do we improve this response. And this is true across cancers. It's not just thyroid cancer. This is a big area of research because although there are certain cancers that respond well, so higher percentages of responses, like in melanoma lung cancer, for example, and others, but the majority of patients may respond and then become resistant. So this is a huge area of study across cancer. And there's a number of reasons that might explain why patients don't respond or why they become resistant, even if they do initially respond. And I've kind of listed those here. We won't go into detail on a lot of them. So one thing that I've been interested in for a long time is understanding what the T cells in the thyroid tumors are actually seeing, because that's a really important component to understanding whether these therapies will work. So the key thing to remember about immune checkpoint inhibitors is that they enhance the patient's existing immune response. You'll hear about other therapies, immune-based therapies, that are engineered. So you're actually making T cells outside the body, express a T cell receptor. You'll hear about that in the next talk as well, that you're engineering the T cells to do their job better. But this type of therapy really depends upon how good the endogenous response is. So I've been interested in understanding what the anti-tumor potential is for patients with thyroid cancer. And a key part to that is understanding what T cells see. So I don't know if you all know the word antigen. An antigen is anything that a T cell can see or a B cell can see. They see their antigen with a molecule called the T cell receptor. And they can see foreign antigens, like in the case of a viral infection or a virus-associated tumor. That would be a foreign antigen. They also can see mutations. So in the case of thyroid cancer, we know BRAF is one of the common mutations. There are others. They're also able to see proteins that might be overexpressed or inappropriately expressed, or tissue specific. So in the case of thyroid cancer, we have certain proteins that are tissue specific and targetable. So one area of work in our laboratory, this is all preclinical, is trying to take these T cells out of the tumors, grow them, and ask what they see. And so this is just showing you a surgical sample that we generate from patients with the help of our wonderful surgeons. We mince this up and we grow T cells out of these tumors, millions of them, hopefully, in most cases. And this is something that I love to look at because it means that the experiment was successful, right? I've got a ton of cells growing here. We freeze those down, and at the same time, we take peripheral blood from the same patient, and we generate these myeloid cells. In this case, these are activated dendritic cells. These are the best cells to stimulate your T cells. And you can see they look right here, that you can see them growing. They are very activated. And our goal is to then put these back together in a tissue culture plate and ask, what do the T cells see? Are there T cells in these patients that can recognize the tumors? Which would be a good thing. That would tell us that we have a bit more hope for these types of therapies. So we can test mutations like the BRAF or RAS mutants. We can test gene fusions, and we can test thyroid-specific proteins. So the goal of this research is, number one, like I said, to assess our potential for immune checkpoint inhibitor success. And also, this could lead to tumor vaccine designs in the future. And that's an area of focus that I would like to pursue. Right now, we have over 15 patients where we have isolated the T cells out, and we're about to undertake studies looking at their specificity. So we've got from low grade, smaller tumors here, to larger tumors with lymph node metastases. And in many cases, I have both primary and lymph node tissue, so I can test both. We also have a couple patients that had undergone therapy and then had surgery post-therapy, DT therapy for this patient, and chemo and pembro, so Keytruda, for this patient. So these studies are underway. The other area of research in our laboratory has been to understand the myeloid component in these tumors. So I said that when you get to a clinically relevant tumor, the myeloid cells look pretty dysfunctional. And so we're trying to figure out, as are many people in the oncoimmunology field, how do we overcome immune suppression by these cells? So this is a image from a tumor in one of our clinical trials, from a patient in one of our clinical trials, just showing you visually how many myeloid cells are there. So the tumor are the cells that are kind of in this grayish white, you can see throughout. The blue cells, which you may or may not be able to see projected here, are sparse. These are T cells. So they're in that tumor trying to do their job. At the same time, we have a large number of myeloid cells, the red, the yellow, the green. Many of these may be suppressive. So you can see in this case, there's just a ton of myeloid cells there trying to inhibit the T cell response. So how do we get rid of these cells? How do we make them better at their job, overturn their suppressive nature? We started out, this was a while ago now, trying to figure out what kind of combination therapy we could use with immune checkpoint blockades. And our first thought was, what happens if we combine lenvatinib with a PD-1 inhibitor? And the reasoning behind that, was that lenvatinib not only it's great for DTC tumors, it's approved, that's your standard of care for patients that are radioactive iodine refractive cases. We knew that it would target the tumor, largely due to its anti-angiogenic effects, so reducing the blood supply to the tumor. But we also know that this drug could overturn some myeloid suppression in the tumor. And this is just depicted here. So this lenvatinib targets the VEGF receptor. VEGF is an angiogenic molecule. It leads to blood vessel development, which promotes tumor growth. If we target it, we reduce angiogenesis, and at the same time, immune cells can express those receptors. And if we block the signal, there's a lot of data out there that suggests they should become more activated, less suppressive. So this combination has been tried and published while we were doing our trial as well. In RCC, it's been approved with a really impressive increase in progression-free survival. Also in endometrial cancer with less of an impressive increase, but some increase in survival. And so this was encouraging to us. We now are about to publish the data in DTC. This was a long process. We had 2 cohorts, and I'm just showing you a little bit of the data today. We had 1 cohort, so these are all RAI refractory patients with DTC. The first cohort was naive to lenvatinib. And so they received both lenvatinib and pembro at the same time. And you can see this is our response rate across patients, and they're color-coded based on the type of differentiated thyroid cancer. We include some poorly differentiated in this group as well. And you see an impressive response rate, but keep in mind that these patients have not seen lenvatinib before. And we don't have a control arm in this study. So this was a little difficult to parse out. We know that in the lenvatinib only trial by Schlumberger back in 2015, that the response was about 63%, and they had some complete response. Here we saw about 67% of the patients responding. So not a better response rate overall. But when we look at progression-free survival, and we compare it in green here to what was seen in the Schlumberger study with lenvatinib alone, we think we're seeing some improvement in progression-free survival at 12 months and at 18 months. Now, we don't have a control arm, but this is encouraging. The second group that was included in this study, in our second cohort, were patients that were progressing on lenvatinib. So they had progressive disease when they entered the study. And you can see here we had a number of partial responses, 4 out of 25. The disease control rate is, I think, what we're most excited about with this study. 96, all but one patient did not progress as their best response. When we look at progression-free survival, we see at 12 months, about 48% of the patients are still doing well, 18 months, about 32. And it's hard to know what this means, but in patients that were actively progressing on lenvatinib, we think there's a signal here. The only thing we can compare it to is really the cabozantinib trial that treated patients that had progressed on lenvatinib. And we see a similar response rate and progression-free survival. So while we can be maybe a little bit encouraged, there may be a signal that we can improve upon. It wasn't the slam dunk we hoped it would be. But my focus now is how do we improve it? And what I've learned from this trial is that it's hard to do it in patients. So we've started looking into preclinical models that we think better replicate the patient situation. So we have a couple different mouse models in our lab. I'll just talk briefly about one of them. This is called the TBP model. This is a mouse model driven by the BRAF V600E mutation and the loss of a tumor suppressor called P53. I like this model. We were given this by David McFadden a few years ago, so this model has been around for a while. I really like this model because tumors progress for at least 14 weeks, which is long in terms of mouse tumors. So it allows time for this equilibrium and escape to happen. And so at the time of treatment in these models, we have a well-established tumor with a suppressive myeloid milieu. And so we've published a bit with this model, and we've initially tried to kind of parallel our human study by testing the effects of lenvatinib, anti-PD-1, which is like the pembrolizumab, or the combination of the two. This just shows you what those tumors look like in the mouse. Pre-treatment, these are the larger tumors that have gone through this 6-week time. And then those that have examples of those that have undergone treatment. And what we saw was that we have a lot of small tumors that vary in their progression. So some will grow out quickly, some will take their time. And I think that reflects the fact that there's an immune response in these mice. And the immune response is doing its best to keep that tumor in check. We do see tumors growing out significantly, especially in the control-treated mice. The PD-1 blockade really didn't do much in this model, which is comparable to what we see in patients. Lenvatinib seems to reduce that outgrowth a bit, and the combination was not better. So the other thing that we do with these mice, they get lung metastases, which also models the patient relatively well. And you can see here a picture of a lung met. We also detect by PCR these metastases, and you can see that treatment really does not improve the incidence of metastases in these mice. And the same is true for survival. So this is a bit depressing, except it's a great model to study the next step. So we've now gotten to the point where we are able to ultrasound these tumors prior, so we can track their growth a bit better. And we're testing a number of different combination therapies to improve upon this potential signal that we have in lenvatinib alone, but we know we need something more than just a PD-1 blockade. One big area that we've moved into in our group is to try to characterize both patient samples and mouse tumors by a process called single-cell RNA-seq. And this allows us to look at each individual cell and assess their expression profile of a ton of genes, which really allows us to understand more about what the cells are that are present. And you can see here, this is one of the mouse tumors that would be prior to treatment. In the red, I've circled myeloid cells. There's a huge population of myeloid cells here, all throughout. The red are those that are probably suppressive, and the green are those that might be still trying to boost that T-cell response. So our big question now is, how does the therapy, the next combination therapy that we're trying, alter this immune response, and which populations may remain in the resistance tumors? That will give us a clue as to what to target next. So will we need additional checkpoint inhibitors? Will we need additional, or just to add myeloid targeted therapies, or maybe we'll need both? So I think this is a really important preclinical step that needs to be taken as we move into the next stage of immunotherapies for patients. So with that, I will thank especially Dr. Brian Haugen, who you may have heard speak here. He's been a long-term collaborator of ours. And we do this work with a whole lot of help through surgeons and tissue banking people and immunologists. And I've had a lot of help in funding from Memorial, from a group called ITOG, which I hope you've heard about. And yeah, and I also thank the patients who obviously were essential for this clinical trial to happen. So if you haven't heard about ITOG, please look into them, because it's a great group of such knowledgeable people that are there to help. And the contact information for Dwight is here if you don't already know it. It's a great group. Okay. Oh, shoot. Okay, that's fine. So Dr. Copland was supposed to be next. He was going to talk about CAR T cells targeted against TSH receptor, which is a thyroid-specific protein. And he's unfortunately unable to get on. So I can attempt to handle questions about that as well, but I'll take any questions that anybody has.
All right, let me bring the microphone to you, and you'll want to channel your inner rock star and hold it close to your mouth.
Hi,
Hi.
I am a patient at Anschutz.
Okay.
Also. So anyway, so I'm lymphema, 8 milligrams, stable disease. At what point, what would my second line be? Would it be adding the Keytruda or going to, I'm sorry, I'm also BRAF V600E plus TERT.
Yeah.
So would the second line be just adding Keytruda? Or would I go to the dabrafenib and MEC?
So I'm a PhD and I should not answer that question, but Dr. Iyer could answer that. Do you want a microphone? Or take hers? Okay.
Thank you for the question. I'm Priyanka Iyer. I'm from MD Anderson. I'm an onc-endocrinologist. I see situations like yours where patients come in on lenvatinib. If it's not progressing, then we obviously wouldn't switch you. But if you are progressing on 8 milligram of lenvatinib, BRAF TERT is a very aggressive disease. In that situation, since you've been exposed to lenvatinib, we would first see where you are progressing. That is, if it can be addressed with localized treatments. For example, if it shows up in the lung or if it shows up in the bone. Lung mets. Okay, so if the lung mets are progressing, one option is either you could go up on lenvatinib. If that's not an option due to tolerance, which happens, we could either add pembrolizumab or Keytruda to your treatment. That's why we are waiting that the group please publish your paper. Or we could just switch to dabrafenib, trametinib, pembrolizumab. What we've seen, and just like what Dr. French explained, we do see a more immune expression following TKIs, like with lenvatinib or even with dabrafenib, trametinib. So combination would be helpful. In just pure papillary thyroid cancer by itself, we don't see a very high PD-L1 expression. So if you were my patient and if I was seeing you and if you were progressing on lenvatinib, I would probably biopsy a lesion if it is accessible, look at the PD-L1 expression. If it is high, then probably switch you to dabrafenib, trametinib, pembro. I hope that answers the question. Okay. I had a question for you.
Yeah.
Can I? Okay. Is somebody else in line? Okay. So what have you seen? Like that was a very nice figure with the anti-angiogenics and immune, the circular one.
Uh-huh.
Do you see the same with cabozantinib? I'm just wondering, like, given that cabozantinib is an approved FDA, FDA approved second line agent, do you think doing cabo and pembro would be similar to lenvatinib and pembro?
So the Cabo Nivo API trial has not been published yet, but has been spoken about in abstract form. And I don't know, I mean, I guess I can speak to the abstract form was they see something similar to what we do. And
But they had
don't,
more toxicities.
I think so.
Yeah. So from a
And-
response standpoint, I don't recall what
But the
whether
response was similar.
similar.
And they didn't take out their survival as long as we did.
Okay.
So it would be hard to compare them.
Mhm.
So definitely, this path might be giving us something beyond the MKI alone,
Mhm.
but there's much more I think we need to do. And the great thing about immunotherapy right now is that when I started doing this research, it was tough to convince an oncologist that they should consider the immune system. Okay, so now we have immunologists and oncologists working together, which is wonderful. I didn't know if I would ever see that. And beyond that, within the immunology field, we have T-cell biologists and we have myeloid biologists. And now we're finally seeing them merge as well. So I think the future is bright in terms of forward movement in these types of therapies.
Okay. And I do have Dr. John Copland on the Zoom now. If we can pause for just a moment, I'm going to ask him to unmute to make sure that we are ready to go with his presentation when we get to that point. Can you hear us?
Yes, ma'am. Can you hear me?
Yes, we can hear you. Is your slide deck finished?
Yeah.
Okay. Dr. Copland, we're going to stop sharing Dr. French's slide deck and let you pull up yours. And in the meantime, if there are any other questions here in the room or in the Zoom, please raise your hand and let us know. Okay.
Can you see my slides?
Not yet.
Hi, nice to meet you. I'm Jen Wong. I'm one of the surgeons at MD Anderson. I really enjoyed your talk. I had a question about the slide and in terms of what you are specifically seeing when you give lenvatinib with pembro. What kind of effects are you seeing in the immune environment with Lenvima that's kind of encouraging in terms of the decreasing the immunosuppressive effect?
So most of that data is not mine. It's historical data in other models, but there's a large field looking at VEGF in different ways. So how do you targeting either VEGF with antibodies, blocking, or targeting VEGF receptor with a drug like lenvatinib or others. So I haven't done a lot of that preclinical data, and I wish I had data in our patients, but most of what I have is preclinical samples that generally were taken years before the start of the study. And the only thing that I have that I could study on trial, on treatment, was peripheral blood. And the one thing that we did see that will be published in this upcoming paper is a decrease in some of the early monocyte lineage in the blood. I don't know what it means exactly. And there's other studies out there showing changes in blood with lenvatinib. So on treatment tissue, we do not have. I wish I did.
Do you think the effect, well, if you had to kind of hypothesize, do you think the effect is primarily in the myeloid populations as opposed to T-cell populations?
Probably. Yep. Yep. Yeah.
We do see
lymphopenia.
lymphopenia with lenvatinib. We monitor, I mean, we don't treat it, we just watch for it.
Mhm.
So do you think that could be playing a role in?
Is there an increase in myeloid at the same time?
I can't remember.
Yeah, it's tricky to look at peripheral blood and make a lot of conclusions from that. The ideal, if I could do the trial again, although it's really hard to get patients for good reason to agree to get biopsies on their tumors. And especially with the lung mets, that's a horrible process and you don't get a lot out of it. So those studies are hard. That's why the mouse model hopefully will give us some light on what the next combination should be.
Okay. All right. We are going to move along to Dr. Copland. It is my pleasure now to introduce him. Dr. Copland is a biologist at Mayo Clinic in Jacksonville, Florida, where he leads the Cancer Biology and Translational Research Laboratory. Dr. Copland and his colleagues develop live cells in culture from patient tumor tissues as well as patient-derived xenograft models. These patient-derived models can be used for drug screening to discover new patient-specific combination therapies. And I'm going to ask you to unmute, Dr. Copland. And your slide share is live. We do see it. You are not unmuted yet. If you go back to your Zoom screen. The wonders of modern technology. There you go. Ask. I saw it on the other window. The one with the red. I don't have my glasses on. I'm on top of my head. The tab with the red circle. Yes, that one. And click that unmute button in the middle. There you go.
Hear me now?
We can hear you, and you'll just need to set your slide share up again.
How about that?
You are good now.
Okay. My apologies for not being there. I'm Al Copland, and I'm going to talk a little bit about a novel CAR T therapy that we're developing at Mayo Clinic for metastatic thyroid cancer. And we think that we can treat every subtype except medullary with this novel CAR T therapy that we've directed towards a protein that sits on the outside of the cell called thyroid stimulating hormone receptor, or TSH receptor. And I want to really thank Jena for giving a beautiful talk and introducing the immune system and especially T cells. So I'm probably going to skip over a few slides to make it shorter for more question and answer. And I'm sure there are more questions for Jena too as well. So CAR T therapy is essentially taking a patient's T cells from your peripheral blood and taking your own T cells, growing them in cell culture and multiplying them to millions of cells. These cells are then engineered to attack your cancer. And so CAR stands for chimeric antigen receptor. That is, a protein or antibody is going to be engineered to be expressed on the cell surface of the T cell, and your new engineered CAR T cells will be re-injected into the patient, and those CAR T cells should be able to now find your tumor that is expressing, in this case, the TSH receptor, and specifically kill that tumor. And so this is just, a depiction of the blue would be a CAR T attacking a tumor cell, and so this light blue protrusion is the TSH receptor antibody that has been engineered into the CAR T, binding specifically to the TSH receptor, the red prongs poking out. And once the T cell has attached, it will start releasing substances that will kill that tumor cell. And so CAR T is a fairly new technology. There are 6 FDA-approved CAR Ts in blood cancers, and there have been many patients cured with this. It has about a 40% success rate with regards to cures and durable responses of up to 60%. And these are just some of the examples of FDA-approved CARs. And CD19, again, is a protein that sits on the outside of blood cells. And so these CD19 antibodies are engineered into a patient's T cells to attack chronic lymphocytic leukemia and also ALL and DLBCL and other blood cancers. CAR Ts have been less successful in solid tumors because of there aren't a number of very good cell surface proteins expressed in tumors that are unique to the tumors. Because if that same protein is expressed in normal tissues, your T cells will attack your normal tissues as well. And so we think we're fairly fortunate in the TSH receptor is fairly specific to the thyroid and makes a good target for CAR T for patients with metastatic thyroid cancer. This is a really great video that kind of helps explain how T cells attack tumor cells and essentially kill tumor cells. And here you can see where the T cell in green is releasing cytokines and poisons. They've attached to the tumor cell in blue and releasing poisons and essentially kill a tumor cell. And they can go on to attack other tumor cells and kill those tumor cells. As you know, thyroid cancer is the 5th leading cancer in women in the US, the 2nd in Saudi Arabia, and the most common in South Korea. And as you know, it's a 3-to-1 ratio in women to men. In the US, we have about 43,000 cases and about 2,500 deaths. So my lab has done genomic profiling from patient tumors, and this is how we first discovered and thought the TSH receptor would be a good target. And in the left-hand panel, you can see these brown staining. In the first column is normal tissue, the second is benign, the third is papillary thyroid cancer, then follicular, and then anaplastic. And so the brown staining is showing expression of the TSH receptor in normal tissue or tumors. And as you can see in anaplastic, there's a loss of the TSH receptor expression in anaplastic, and we think that we can cause re-expression of the TSH receptor using MEK inhibitors that I'll show you in a minute. And in the right-hand panel in blue, you'll see very high expression of the TSH receptor in the thyroid gland. There is also expression in the testis, but it's immune privileged and probably will not be affected by the TSH receptor CAR T therapy that we are proposing to do in a clinical trial. This is simply the structure of the TSH receptor. The top half is on the outside of the cell, and then it penetrates into the cell, and the TSH receptor essentially stimulates the production of thyroxine and triiodothyronine, or T3 and T4. It's a G-coupled protein, and as I told you, it's traverses the cell membrane and protrudes outside of the cell, and we take advantage of the part of that protein that sticks out of the cell that we can make an antibody that will bind to it. Chimeric antigen receptors, this is a figure to describe how they're designed, and as I said, on the extracellular part of it, just looking at the first green figure to the far left, the extracellular part is an antibody that recognizes the protein on the cell surface of the tumor, and the middle part is essentially once the antibody has bound to the cell surface of a tumor, it sends a signal through this CAR to activate the T cell to kill the tumor cell. And so this transmembrane domain and intracellular signaling pathways made up of proteins like CD28 and can activate the T cell. This is simply the amino acid sequence of the TSH receptor monoclonal antibody that we're using in our particular TSH receptor CAR-T. These graphs don't mean much to you, but as you can see in the red line going down, as you increase the number of CAR-T cells, this is in cell culture, co-culture of TSH receptor CAR-Ts, in co-culture with tumor cells, and you can see that as you increase the number of these TSH receptor CAR-Ts, they're killing the tumor. On the right-hand side, these are cytokines that are secreted from the CAR-T, indicating that it is active and killing tumors. And so cytokines like interleukin-2, GM-CSF, interferon gamma are shown to be upregulated in these activated CAR-T cells. We've also grown tumors in mice, and this is the gold standard that allows being able to show that we can inhibit tumor growth in mice, will allow us to move on to clinical trials. And here we've injected about 5 million or 11 million tumor cells underneath the skin in the mice. These tumors grow very big, and in the top right-hand corner, you have the number of days that these tumors are in mice. And so we go out to day 20 and 21, and the left-hand y-axis is tumor volume. So as these lines increase, it shows you the size of these tumors. And so in the green line is the control. And so within 15 days, these tumors are about 1,200 cubic millimeters in volume. And you can see with 5 million cells, we can suppress that tumor growth, but then they do finally escape. And with the 11 million cells in red, we can keep these tumors suppressed. And in the far bottom right-hand corner, we can do immunohistochemistry on the tumors once we remove the tumors from the mice. And you can see this is brown staining is human CD3, showing that these T cells have infiltrated into the tumor and are killing tumor cells within the tumor. One exciting finding that we have found is in Hürthle cell, which has been renamed oncocytic thyroid cancer. And we examined a panel of Hürthle cell patient tumor tissues for expression of the TSH receptor. And we looked from very low grade to very high grade tumors, and we saw expression of the TSH receptor in every Hürthle cell patient tumor tissue that we examined. Importantly, there's no FDA approved therapy for the treatment of Hürthle cell. So we think that our TSH receptor CAR-T therapy might be a very effective therapy in patients diagnosed with metastatic Hürthle cell thyroid cancer. This is in an animal model. Again, we're looking at tumor volume, and the blue line is the control. And you can see that over about 50 days, you can see the increase in tumor volume to about 1,500 cubic millimeters. You can see that if we inject the TSH receptor CAR-T, that we completely suppress the growth of these tumors. We then re-challenged the tumors at day 51 with new Hürthle cell tumors. So we put another 10 million tumor cells into the mice that had been already treated with a single dose of this TSH receptor CAR-T. And if you follow the red line in the second graph to your right, it shows that these tumors are completely suppressed, and also the new tumors that we put in are completely suppressed. So the CAR-Ts are still present, they are reactivated, and they start multiplying again to kill these tumors. And so this is an example of a potential curative therapy for patients with Hürthle cell, metastatic Hürthle cell thyroid cancer. As from the introduction, you know that my laboratory makes these patient-derived xenografts, so we have a number of these models that you can see in the table. A lot of these are anaplastic thyroid tumors, and as I showed you earlier, anaplastic thyroid tumors lose the expression of the TSH receptor, and this is through a MAP kinase ERK pathway that gets overexpressed as these tumors become dedifferentiated. And so we can essentially use MEK inhibitors that are actually FDA approved for BRAF mutant tumors, and we can use MEK inhibitors with or without BRAF inhibitors such as dabrafenib and trametinib and cause re-expression of the TSH receptor. So you can see here in the right panel, we've treated mice with dabrafenib and trametinib, as well as this RO5126766, which is a MEK-RAF inhibitor. And you can see that, my apologies, you can see the positive control that's got nice brown staining in the far left panel. In the second panel, the placebo control, there's no TSH receptor expression. And after a week of daily treatment with dabrafenib and a MEK inhibitor, you can see brown staining in both of the two panels to the right. So we re-express the TSH receptor. So the question now becomes, can we do a combination therapy with MEK BRAF inhibitors plus our TSH receptor CAR-T? And the answer is yes. Here, again, we've treated either with the control in the line that goes to about 1,000 cubic millimeters in dark blue, with the TSH receptor CAR-T by itself in red square boxes. You can see a small amount of inhibition. So again, this indicates that the TSH receptor is not being expressed. The MEK and BRAF inhibitors in the light blue line and the upside down triangle shows strong anti-tumor activity, but when you combine the MEK and BRAF inhibitors with the TSH receptor CAR-T in the bottom red line, with you can see complete suppression of tumors. So in patients with anaplastic thyroid cancer, we believe that we can cause re-expression of the TSH receptor, allowing us to then treat patients with CAR-T therapy. So we'll definitely need a companion diagnostic that we're developing to determine whether we can re-express the TSH receptor in patient tumors. And so, again, this is sort of summarizing. We need to be able to express the TSH receptor, a number of metastatic thyroid tumors. These are patients that are radioiodine resistant and have no other options for therapy or resistant to other therapies. And so, a number of these tumors, such as follicular thyroid cancer, classical papillary thyroid cancers, or Hürthle cell or oncocytic thyroid cancers that continue to express the TSH receptor, this CAR-T therapy would be very ideal and potentially curative for these patients. And with anaplastic thyroid cancer or poorly differentiated thyroid cancers where we've lost the expression, we can potentially do combination therapy with MEK BRAF inhibitors plus the TSH receptor CAR-T. This is a busy slide, but essentially is a clinical trial design that we have planned and hope to have up and running within a year to year and a half. We're currently trying to get through FDA approval with a new with investigation of a new drug, and also develop the GMP manufacturing of this CAR-T. And this is the clinical team, myself and Dr. Saad Kandarian. Saad is the expert in thyroid and in CAR-T therapy and has designed these CAR-Ts. Dr. Mabel Ryder, which most of you will probably know, will be the PI on this clinical trial. And Dr. Yuji Zhao and Mohammed Karfan Damaja are on the Florida campus with me and would be the clinicians running the trial on the Florida campus. And Hunchin Chai and Dr. Parent work with me on the Florida campus developing companion diagnostics, and Michael Rivera is our thyroid cancer pathologist who's developing immunohistochemical companion biomarker as well. And Dr. Han Chen and Cynthia Wilkins and Tim Wiltshire are part of the Mayo GMP production team that will be producing the virus and the CAR T therapy for the patients. And so I'll stop there and take any questions that you might have, and like to thank each one of you for taking your weekend to really be present and to try to help all of us cure every thyroid cancer that's out there. And this is one of my favorite sayings that Margaret Mead, and essentially, it's never believe that a few caring people can't change the world, for indeed that's all whoever have. And each one of you today that are here are those people. So thank you.
Thank you very much, Dr. Copland. I'm going to start with an online question, and then we have a question here in the room. The first question is there some hope that CAR T therapy could work with oncocytic Hürthle cell? Dr. French, do you have thoughts about this that you'd like to share? I'm not sure.
Yeah, so I think what he showed was that TSH receptor was expressed on the patients that he had looked at. So certainly there's hope. With CAR Ts, the hematology field, with CAR Ts strategies, it began, as Al said, with the hematological malignancies, and it's still, they had great successes, and they're about 10 years ahead of the solid tumor field. And what they've been working on is how do you keep those T cells alive long enough? And then what happens if the tumor outsmarts the therapy, right? And so they are ahead of us, but they have multiple markers, and they're trying to do dual therapies that target more than one marker at a time. And so that's where the solid CAR T field will have to get eventually. But the fact that we have, I think, 3 CARs right now in the thyroid cancer space is really encouraging. And that initial data will help determine where we have to go in the future. But the hematological field has the biology of the CAR T, the mechanics of it, how it signals. There's so much study going on to make it better. So we're stepping in, I think, at the right time to really take advantage of what they're learning. So yes, I think there's a lot of hope in all cases.
Is there some hope that CAR T therapy could work with oncocytic Hürthle cell? And Dr. French weighed in as well.
The answer is yes. We think. Can you hear me?
Yep.
Yes, we can.
Yes. We can. Yes. If the TSH receptor is there, we think it will be curative.
Okay, wonderful. And here we have a question in the room.
Thank you, Dr. Copland, for such a lovely talk. This is Priyanka Iyer from MD Anderson. I'm one of the oncologic endocrinologists. Hi. So I treat advanced thyroid cancer, and my question is, actually I have 2 questions. One, what is the half-life of the CAR T cells that are infused? How long do they circulate? And number 2, you showed about the redifferentiation therapies following MEK inhibitor or dabrafenib and trametinib and expression of TSH receptor. Are we thinking of giving the CAR T infusion right after, or do we wait?
So, Priyanka, the first question was, what's the half-life of the T cells? We measured the T cells weekly in the blood, and we know that they're highly replicative up to 3 to 4 weeks. The data that you saw in the Hürthle cell experiment was, the key experiment where we've treated the animals, the tumors regressed, and then we re-challenged those mice that had been treated with the TSH receptor CAR T, and you can see that those new tumors that were put into the mice were killed, and those were 10 million cells. And the graph on the right showed that these cells were, that the CAR T cells were still present. They were reactivated to multiply again, so they made new CAR Ts, and that could be measured in the blood. So we think, go ahead, Priyanka.
So I was just wondering, sorry, I didn't mean to interrupt you, but my thought raced ahead. So that means, does it stimulate the patient's body to make more CAR Ts, or is it just that it's lasting long enough?
Jenna, you're
I know
welcome to answer.
Yeah.
Answer that. Well, I mean, oh, shoot, I think I just turned it off. Can you hear me? I mean,
Yes.
I'm not a CAR T cell expert, but remember, he's working in mice that don't have an immune system. The
Oh.
patient has to be lymphoablated. So it's not a full, it's not like a bone marrow transplant ablation. As far as I know, it's a low level ablation, but your immune system, they want it to come back so that you don't get sick from not having an immune system. Does it echo? But there's a lot of studies that have been done on how long the T cells last. And then there's even more work being done on how do we get them to last longer? Do you add a checkpoint inhibitor? Because that's one thing that happens, is that the CAR Ts become tired from trying to do their job. They upregulate PD-1. And so do you add a checkpoint inhibitor to make them live longer? Yeah, that's my 2 cents on that.
And I've forgotten the second question, Priyanka.
Yes, sure. So my second question was, in the clinical trial design that you showed, where there was redifferentiation with MEK inhibitor followed by CAR T, would you be thinking of giving the CAR T infusion only if they progress after the redifferentiation, or right after the redif treatment radioiodine is given?
Well, these patients are going to be RAI refractory, and these patients will also potentially be drug resistant to other therapies. And so we've got some of our anaplastic thyroid cancer models. We've been able to, we typically treat these models daily for a week to re-express the TSH receptor, and then we envision checking that either with a biopsy for TSH receptor positivity, or we're also developing a radionuclide diagnostic that would be much less invasive than a biopsy to demonstrate the TSH receptor has been re-expressed in the patient's tumor before administering a TSH receptor CAR T.
Understood. My question is, like, when you do the redifferentiation therapy with Mekinist, these tumors might redifferentiate to now be RAI avid. So would you treat them with RAI and CAR T after the redif therapy, or would you just do the RAI and only if they progress despite RAI, you would do CAR T?
Got you. Oh, I got your question now. My apologies. So, and that's, yeah, and that's occurring in a number of trials with regards to this clinical trials, and part of the issue is sodium iodide symporter, which has to be re-expressed to be other parts of the other genes have to be re-expressed, and not always does the sodium iodide symporter come back with or without the TSH receptor. The TSH receptor can regulate and upregulate NIS, thyroglobulin, and other genes that are critical for iodide uptake and retention into thyroid cells. But our intent would be, if the TSH receptor is present, we think this is curative, that we would move forward with the TSH receptor CAR T and not try to re-challenge with radioactive iodine.
Thank you.
Great question, and thank you for that question.
And Dr. Copland, I know that you are in a time crunch. We have 2 more questions from our online audience. Do you have time for that?
Yes, ma'am.
Okay. Tracy asks, how far away is CAR T for oncocytic in human trials?
We hope to be in trials in early 2025. So we're pushing as hard as we can. We're at a pre-IND setting right now. So you've got to file an IND, investigation of new drug, with the FDA, and the FDA has 30 days to respond and approve or disapprove. Where the biggest holdup is in the manufacturing process of CAR Ts. You've got to get GMP grade plasmid, and you've got to get GMP grade virus, and that's sort of our biggest hurdle right now. And so we've been pushed back about 4 or 5 months because of the company that's not done what we ask them to do. So otherwise, we'd be moving a lot faster.
Okay, thank you very much. The next question is 2 parts and a comment. With the highly targeted TSH receptor, will there be less risk for adverse events and or damage to normal cells? That's the first part. The second part is also, how do you check that a patient's TSH receptor is intact? Is this measured in the blood?
Right. So what was the first question again? I'm sorry.
That's okay. With the highly targeted TSH receptor, will there be less risk for adverse events and or damage to normal cells?
Yeah, and the answer is yes. And that's the reason we chose the TSH receptor. We've not had any adverse events in our mice to date yet. So and again, the TSH receptor is highly expressed in thyroid and very little in other tissues. And what was the second question again? I'm sorry.
How do you check that a patient's TSH receptor is intact? Is this measured in the blood?
It's not measured in the blood. It has to be intact and expressed on the cell surface of the tumor cells. So we would propose to do a biopsy of the patient's tumor tissue to demonstrate the TSH receptor is present. And like I said, we're also developing a radionuclide diagnostic, which would be a radio-labeled TSH receptor. It's very similar to radioactive iodine that's used currently to test for iodide uptake in a patient's tumors, indicating that they would be a candidate for radioactive iodine therapy. So we're developing a radionuclide specifically for the TSH receptor that would allow us to inject this into the patient's blood, and it would then show that the tumors either do or don't express the TSH receptor. So it would be very similar to a PET scan after radioactive iodine injection.
Thank you. And then the last part of that is a comment. Thank you for giving refractory oncocytic carcinoma patients hope for the first time. Thank you. And with that, I will join the folks in the room here with thanking you and Dr. French for your time today.
Yes,
Thank
I
you
wish I
very
was
much.
there. Yes, I'll do my best to be there in person next year.
And that concludes our session. Thank you so much for your time.
Okay, thanks, Jenna.
Thank you.
Bye-bye.
Bye-bye.